Hi everyone. Let me tell you about the types of hemolysis shown by bacteria on blood agar. The common types of hemolysis shown by bacteria are alpha hemolysis, beta hemolysis, gamma hemolysis, alpha prime hemolysis and target hemolysis. What is alpha hemolysis? It is a green zone seen around a bacterial colony which is shown by catalase negative bacteria. Due to the lack of catalase enzyme, hydrogen peroxide accumulates near the bacterial colony. This hydrogen peroxide oxidizes oxyhemoglobin to methemoglobin, which appears green in color. Some books explain alpha hemolysis as partial hemolysis, but the actual mechanism is formation of methemoglobin. There are no hemolytic enzymes involved in this reaction. The bleaching of alpha hemolytic colonies on chocolate agar is also due to the formation of methemoglobin. Alpha hemolysis is shown by catalase negative organisms like Streptococcus pneumoniae and the viridans group of Streptococci, which is a huge family consisting of many organisms. I'll just give the name of some organisms like Streptococcus aeruginosus, Streptococcus mutans, Streptococcus sanguinis, Streptococcus oralis, Streptococcus gordoni, Streptococcus mitis, Streptococcus salivarius, Streptococcus vestibularis, Streptococcus sinensis, Streptococcus intermedius, Streptococcus sobrinus, Streptococcus parasanguis, Streptococcus crista, etc. Now, what is beta hemolysis? It is a clear colorless zone seen around a colony of bacteria. Here, red blood cell membrane is ruptured. It is due to the action of enzymes called hemolysins. In Streptococcus pyogenes, which is catalase negative, it shows beta hemolysis due to the enzyme streptolysin S. Now, what is gamma hemolysis? Here, there is no action on red blood cells, that is no hemolysis. What is alpha prime hemolysis? A small zone of intact erythrocytes are seen immediately adjacent to the bacterial colonies with complete hemolysis surrounding the zone of intact erythrocytes. This type of hemolysis is shown by some species of Streptococci and the exact mechanism has not been explained in literature. Now let me tell you about target hemolysis. Also called double zone hemolysis because it resembles a target which is used for shooting. Clostridium perfringens, when grown on anaerobic blood agar, produces an inner zone of complete hemolysis due to theta toxin, surrounded by a wider zone of incomplete hemolysis due to alpha toxin. In Clostridium perfringens, the hemolysins are called toxins. All enzymes, that is hemolysins, whether it be alpha hemolysin, beta, gamma, delta, theta, whatever it be, they produce only beta hemolysis of varying intensity and varying pattern. Let me give you some examples of hemolysins produced by bacteria. Hemolysins of Streptococcus pyogenes include streptolysin O and streptolysin S. Oxygen labile streptolysin O is an antigenic extracellular protein. So antibodies are formed and you can get anti-streptolysin O, that is ASO titer, which indicates the infection by the organism. Hemolysis can be demonstrated only in anaerobic conditions or by stabbing into blood agar at different places. Streptolysin O binds to cholesterol on cell membranes, forms pores on the blood cells and ultimately lyses it. Apart from red blood cells, they can also lyse leukocytes, monocytes and culture cells. Hemolysins similar to streptolysin O include listeriolysin O, which is produced by Listeria monocytogenes, tetanolysin, which is produced by Clostridium tetani, theta toxin of Clostridium perfringens and seriolysin produced by Bacillus cereus. The second type of hemolysin produced by Streptococcus pyogenes is an oxygen-stable streptolysin S, which is not antigenic and it is seen inside the bacteria. It is called hemolysin S because it is soluble in serum. Serum is essential for its expression. It is released by a carrier agent like serum albumin, RNA, or alpha lipoprotein. Here it interacts with the membrane phospholipids of red blood cells, causing swelling and lysis of the cells due to disruption of the osmotic barrier and leakage of ions from the cells. Here also, apart from red blood cells, the polymorphonuclear cells and platelets are also lysed. Now let's discuss about the hemolysins produced by Staphylococcus aureus. Alpha, beta, gamma and delta hemolysins are produced. All these hemolysins produce beta hemolysis. Let me tell you about alpha hemolysin. Alpha hemolysin produces beta hemolysis, which we see commonly around the Staphylococcus aureus colonies on blood agar. This property is best seen with rabbit blood agar. They lyse RBCs and polymorphonuclear cells. It is a protein which is secreted into the medium and they form pores on the cell surface which causes efflux of potassium and other small molecules and influx of sodium and calcium which causes osmotic swelling and rupture of the blood cells. Some Staphylococcus aureus produce beta hemolysin. A beta hemolysin producing Staphylococcus aureus strain is called Plasson strain, that is ATCC 25923. This strain is used for a test called CAMP test. I have made a separate video on CAMP test. You can watch that to get the details. The beta hemolysin produced by Staphylococcus aureus is a sphingomyelinase. It is an extracellular protein and the hemolytic property occurs only in the presence of magnesium ions and it acts only on cells with sphingomyelin and lysophosphatidylcholine. Sheep blood is rich in both sphingomyelin and lysophosphatidylcholine, hence it is ideal for the action of beta hemolysin. The beta hemolysin also shows a hot-cold phenomenon. That is, the hemolysis starts at 37 degrees Celsius but gets enhanced on chilling. Initial disruption of cohesive forces within the cell membrane occurs first, and when the temperature is lowered, phase separation within cell membrane occurs leading to lysis. Staphylococcus aureus also has gamma hemolysin, which is an enzyme that acts by forming pores on blood cells, thus altering the cellular permeability to potassium and other cations and rupturing the cells. Delta hemolysin of Staphylococcus aureus acts like a surfactant that disrupts the cell membrane of blood cells. Hemolysis of Clostridium species are also called toxins: alpha toxin, delta toxin and theta toxin. Here it is the alpha toxin which gives the reverse CAMP test, which has also been discussed in detail in the video on CAMP test. Gram-negative bacteria like E. coli, Aeromonas, Pseudomonas, etc. also produce hemolysins. Now, what is the difference between hemolysis and hemodigestion? Hemodigestion means some bacteria like Vibrio cholerae produce some metabolic byproducts which cause a non-specific destruction of blood cells. This may be observed more around confluent colonies and may not be seen around individual colonies at times. I guess by now you have a clear idea about the types of hemolysis shown by bacteria and the mechanism behind this property. Thank you.